Population genomics. Population genomics is the study of genomes of a specific population of specific strains, bacterial strains, or the varieties or organisms. So in case of a population genomics, we study the genome of a specific population. We study the genome of specific strains, that can be the bacterial strains. We study certain varieties, genomes of certain varieties, or the genomes of different organisms. So such a study or such a subdiscipline of genomics is called as population genomics. In population genomics, we study about the genetic diversity which is present between the population of a specific location. Specific population can be the human population. If we study the genetic diversity between the genomes of the humans, that would be the example of population genomics. Population genomics helps us to understand disease pattern and new insights about the response of the drugs with relevant to specific individuals. Or we also study, and it is also helpful for the humans, that how the drugs they respond to specific individuals based on the genome of that particular individual. Population genomics has given deeper insights about the genomic size. It has elaborated further complexities of the genomes of the organisms, and it has identified various genetic variations which are present between the individuals of a specific population. Like if we take the example of the humans, population genomics, human genome is approximately 200 times greater than the yeast, while it is 200 times smaller than the amoeba. So amoeba is a unicellular animal, while humans, they are multicellular, and yeast is also multicellular as well as eukaryotic. So if we do the comparison between the genomes of these three, we see that human genome is larger than the yeast, while on the other hand, it is smaller than the genome of the amoeba. So less than 2% of the human genome is coding. So population genomics, when we have performed the population genomics, we also understand that what is the percentage of the genome that is coding or non-coding. There are two best examples of the population genomics. One of the best example of population genomics is 1000 genomes. 1000 genomes was actually a project in which the genomes of 1000 individuals were sequenced to determine the variation at the DNA level between 1000 individuals. That is the reason it is called as 1000 genomes. As a result of 1000 genomes, detailed and medically useful picture of the human genomes came into being, and with the help of 1000 genomes, all those rare genetic variants were identified as a result of 1000 genomes. As a result of 1000 genomes, it was evident that any two human beings, they are 99% identical at the genetic level. Genetic variations which were found as a result of project of 1000 genome has revealed that susceptibility to the diseases as well as response to the drugs. So based on the picture of 1000 genomes, now we are more evident and we are more familiar that which are the genetic variants due to those genetic variants which individuals that are more susceptible for certain diseases. The second example of population genomics is HapMap project. HapMap project has also discovered many regions on the human genome which are associated with many diseases, like the most common diseases can be the cancers, diabetes. So HapMap project also has identified and revealed those variations. If an individual has that particular variation, genetic variation, that individual would be more susceptible for those diseases. These are the two examples of population genomics in international HapMap project as well as 1000 genomes. So these are two best example of the population genomics. So what were the goals of population genomics? The actual goals of the population genomics were to produce a catalog of variants that can be 1% or greater frequency in human population, and to identify these variants should be up to 0.5% within the genes, and increase the sensitivity of disease discovery was one of the goal of the population genomics in case of HapMap and in case of 1000 genomes. Population genomics also provides better understanding of every rare genetic disease. With the help of population genomics, we can understand the contribution of common variants to the common diseases like diabetes and heart diseases. One of the goals of the population genomics is also to identify SNPs, but at a large level, like rearrangements, deletions, and duplications. Means population genomics also helps us not only to identify the genetic variant at the single point, but with the help of population genomics, we can study the chromosomal rearrangements, we can study the chromosomal deletions as well as duplications.